Okay, great. So thank you guys for coming today. Um, you would have Matt Scholz, the founder and CEO of Oisín Biotechnologies. I always love the name, by the way. It's a longevity clinic medicine company that targets cells via DNA-targeted interventions. And the targets are those implicated in aging, such as senescent cells, muscle cells, and adipocytes via their proteolipid vehicle platform. Matthew's been part of Ocean for a long time now, and the name, I believe, I did a bit of research, but I think it comes from some Irish mythology. I would love to hear about that too. And you started the company as a student, so just like most of us here, studying computer science at the University of Washington. So today, Matt is gonna talk about Ocean a bit, give us some career advice for those here who are interested, and yeah, and to answer some questions that we have. So thank you, Matt, so much for joining us.
Yeah, my pleasure. Well, oh yeah, and Oisín was as a Gaelic myth. Oisín is the name of a person who journeyed to the land of eternal youth off the coast of Ireland, and there's a whole bunch of permutations about it. But I can't take credit for the name. My co-founder had been saving it for a very long time. He's like, "We got to call it Oisín," and I said, "Well, if the domain name's available, sure." And that's about all the influence I had on it. Well, awesome. Well, it's great to be here and to chat with you guys. And what I've done is actually I put in kind of a random assortment of slides that doesn't just cover what Oisín itself is up to, but kind of the journey I took into bio, because it seems like that's probably relevant to people here. So, like as Lisa pointed out, my background is actually originally in computer science and physics. So I did a bunch of cybersecurity stuff, and that led me down this very strange path. And I always think it's a fascinating topic. How do you start a company in a space where you like don't belong, kind of, as you're building something totally outside your trained expertise. And I think that a lot of the greatest innovations happen when disciplines mix. And so it's a really exciting place to operate, but it can be a challenging one as well. So I've been told I've got like 15, 20 minutes to talk about slides and whatnot, and then go to Q&A. And there's not a lot of people here, so you guys feel free you can interrupt. We don't have to go through just a thing of you listening to me, but I'll take you on a journey that hopefully will entertain everybody. And so I guess before you can get into all the fun stuff, it's worth kind of, I guess, contemplating where we've come from in the sense of medicine. And I think for most of human history, medicine was, plants and prayers, basically. Small molecule drugs and rituals, and it stayed like that, and in fact it still is a large part like that. Medicinal chemistry is still a very dominant modality, and a lot of the drugs we have are still derived from plants. But it wasn't until relatively recently that people started playing with other kind of more advanced modalities, and some of them, they came surprisingly early and were surprisingly crude but effective, like the first of, like the live biologic vaccine is the cowpox vaccine, where they're taking like the stores from the cowpox and rubbing it on people. And there is a funny history that there's a term still, roughly in English about, having skin as smooth as a milkmaid's. And the etymology of that was the milkmaids were frequently exposed to cowpox and had developed a kind of an immunity to smallpox, and so they were frequently missing the scars that plagued so many people who when smallpox was more endemic. But these like really early things, and even actually the first immunotherapy that we think of for like cancer, it's like the checkpoint inhibitors and all these things. That there's an ancient predecessor to it that's crazy if you think about it, where they would actually take someone who is really sick from cancer, and they would put them in a bed of someone who had died from smallpox, or occasionally inject them with bacterial solutions. And these things, like a 50-50 cure-kill rate, but they would cause such a bad infection that the immunomodulatory shield of the tumors could fail, and you'd have like remission from it. It's pretty amazing if you think about it, given how virtually nothing about why that would work. But fast forward to like the rise of recombinant biologics and things like insulin, and then cell therapies with early bone marrow transplants only from really the 50s, and the first approved cell therapy being, after 2000. And but the logical, I guess, next step is to begin modifying the DNA in these cells, something that people were interested in ever since they knew what the double helix was, but only fairly recently gained the ability to do it in controlled manners. And I suppose if you're looking at where things are going, this would be a controversial point for sure, but I think the future of humanity is that we effectively take control of our genetic fate, that it's effectively inevitable that humans, in a few hundred years are going to be the same as we are today, and that they're going to be changed. And of course, the first documented genetic modifications, germline modifications, so human embryo, were done in China back in 2018. And so all that to say, when I was getting into my journey, I started a company called Immusoft, and this was before there were any approved gene or cell therapies. And I was initially trying to use cryptographic hardware, like password crackers, to evolve the immune system in silico. I wanted to make new kinds of broadly neutralizing antibodies, and they could effectively wage a war against a virus with computers instead of flesh and blood. And as I embarked on this path, it was pointed out to me at some point that cells don't have USB ports. You can't just make new code and upload them. And I thought that was absurd. And so I started this company called Immusoft, which a portmanteau of immune system software, to build what amounted to as an app for the human body, more specifically producing autologous long-lived plasma cells that would secrete therapeutics. And the thesis for this, and really all the other projects I've been involved with since I got into the space, was that the essence of life is information, and chemistry is a substrate. But if you can manipulate the code in life, you can do anything. And so in this case, like a long-lived plasma cell is a crazy prolific secretory cell. Like a mature one can make between 10 and 100,000 antibodies every second. And you've got 10 to the 10th of these things in your body. And so there's enough actually circulating immunoglobulin in your blood that if you took it out and spun it in a centrifuge, you could actually see it. I mean, assuming you didn't die from losing all your blood, but there's a lot of it in there, grams and grams. And so we're basically trying to hijack that machinery to make drugs. And this was partly based on this idea that I wanted to recreate the biochemical environment of youth as you age, to like put back all the things that go missing. Like if you took your blood, once a year from birth till death, it's not the same, right? Things change inside as you age, and you think about like obvious things like say estrogen or testosterone, but countless other things do too. And you can never take enough pills to like undo this. But if you could just upload new programs to your body, maybe you could put it back. And so it's kind of a wild idea, and it ends up actually going into clinic. So this, not for that, but for a rare disease called MPS1. It's a genetic disease that affects children. And this is remarkable in that it's the first engineered B cell therapy to ever go into a human. And so in this case, the patients have a genetic defect. They can't make an enzyme they need to survive. And it's really nasty. Like untreated, you melt down from birth. You're not going to live much past 10. The standard of care is a stem cell transplant when you're about 18 months old, and it still has like major problems as a youth. And so they've roughly managed to double the average lifespan, but it's still pretty bad. And if you are getting enzyme replacement for it, it's hundreds of thousands of dollars a year, every week. And like I said, it's not curative. And so we had basically modified their immune cells to make this enzyme 24/7 and just dump it into their blood. And so it was like the first of these projects that we ever made. It took 12 years to go from idea to clinic too. It was a fairly painful journey, but I think that's one that maybe you have to be prepared for if you're trying to build new classes of treatments or go after really ambitious goals. But one thing, and it's kind of a bit of a sideline, but I think it's interesting, it probably is interesting to this group, is that when I got into bio and I had to, make my own lab, hire a team, all this stuff from scratch. And it really became apparent that like the one person in medicine who has no say in anything is the patient. That, if doctor won't give you the drug, you're screwed. If it's not approved, you're definitely screwed. Insurance won't cover it, you're out of luck. And this is something that's only become more of an issue as therapies have advanced. I mean, if you go back to our plants and prayers, like you can make a lot of small molecule drugs in a fairly primitive lab, and you can order a ton of compounds on Alibaba without many questions. But you can't do something like make an antibody. You couldn't make Herceptin in your lab. Even if you had like $1 million lab, like a decent space, like you're probably gonna kill yourself. And so I got really interested in trying to shift the balance of power in medicine back to the patient so that you could effectively do things like gene therapy in the developing world or in the garage effectively. And I mean, you think about the developing world's a whole other issue, but I mean, even if we made these things for free and said, we'll just give them to you, like most of these places couldn't pay for postage. I mean, you need a cold supply chain to distribute them. And that's really prohibitive when you have to do infusions of something every month. And but there was a really fascinating confluence of technologies that had occurred in this time, and that's that the cost of DNA had been falling faster than Moore's law. Like right now, you can type a gene sequence into your iPhone; it'll show up in the mail, FedEx envelope. Write a program in the language of life, get delivered to your door. And if you run a electric field across the cell, you can put nearby things inside of it. And it's this technique called electroporation, and it's used in the lab all the time. It's super common. And they basically are tasing cells in their dish. And the electric field will punch a hole through the membrane of the cell. DNA is a slight negative charge. You can drag it right into the cell. And this is used all the time, but what's fascinating is the same thing actually works in living tissue. So you can electroporate a mouse or a person, and this here is showing an intramuscular electroporator, which is considerably less pleasant than one that's just on your skin. But in this case, these needles, go about an inch deep, or I think 25 millimeters long, into the muscle. You put DNA in the middle of it and effectively plug it into a light socket. You'll get a little, cubic centimeter drug factory that will make whatever you want. And so we had actually done this during the Ebola outbreak a few years ago with in monkeys, where we got the antibodies, the sequences for ZMAP, and we modified them for mammalian expression. And we grew up all this DNA in the lab, like Breaking Bad style. And it's like tons and tons of gator preps. And injected these into monkeys. And for a few $100s worth of DNA, we made a treatment that would cost more than $100 grand. And I think that's like a really powerful technique when you start thinking about how could you, level the playing field in terms of development and access to advanced therapies. And there's already a bunch of like commercial in vivo electroporators out there, and they, for the most part, won't let you play with them. And so I got very interested in actually has a cool little image before I forget. So this is putting just GFP into the muscle. It's just like a mouse, but and you can see the pattern from the GFP expression from it, which is pretty crazy. But so you can do that in a person though, and you can do it with very primitive technology. And so I was interested in making an open source in vivo electroporator that could just publish to the world, so people could cobble these things together and do whatever they wanted with it. And what was odd is as I went down that path, I got eventually kind of over my head in electrical engineering on it, and I was trying to make it solar powered so you could charge it with a solar cell, and it takes like a pretty high current discharge to work, and so that I was having trouble with it. And I ended up connected with this guy, Chris Peel, and he's a pulse power expert, but he's the CTO of Helion, a fusion power company that's initially backed by Y Combinator, and actually quite successfully, and I think a year or so ago, Sam Altman gave him like a $500 million personal investment. It was like a crazy operation. They have a functioning fusion reactor sitting here outside Seattle, which is nuts. But he had asked, like, why don't you use a magnetic field instead of the electricity? Like, why are you using current for this? And or point to point current. And I was like, well, that's much harder. I'm already in over my head at the simple one. And he's like, okay, well, I have some ideas. And he basically cobbled together a prototype and showed up at my lab in the middle of the night. Where like put some blood into this with some just GFP DNA, and we're like, how many times do you hit the button? Basically, so push it a few times, and then put the cells in the incubator, and so we'll let the scientists isolate this in the morning and see what happened. And And what is really sweet is able to transfect PBMCs in whole blood, something that you can't do with an electroporator. And it led to the formation of this company, Sigma. And at the time, we didn't have time to be starting more companies. And so we took it to Y Combinator and brought on a CEO and turned it loose. And it's become this well, a rendering of it. The real one's scarier looking. But this device that you can use to make things like CAR T cells with less cell death. And what's really crazy about it is it works in red blood cells. And so in this case, the idea is kind of like, you think like an electromagnet, or have you ever seen these things where you like spin a magnet next to a wire that's attached to a light and the light will flicker if you spin it. And so magnetic field changing induces current, a stable magnetic field does not. And so this thing is a little bit like going through an MRI at Mach 2. Like it's a soul-crushingly powerful magnetic field that comes on like 1,000th of a second and then goes away. And so it will transiently induce a current across it. But since it's not a point-to-point current and it's not sustained, there's no risk of like arcing or the thermal stress. And so if the electroporator, if you put up the voltage too high or the buffer's not right, you get, what amounts to a little lightning bolt in your dish or your muscle, and that's a bunch of barbecued cells. And so a lot of electrophorators are relatively good at getting things into cells, but they often a lot of the cells that were there at the beginning cease to exist. And so they just get completely obliterated by it. And with this approach, you don't have that happening. And strangely, it can put things into red blood cells. And so its latest project has actually become a thing with the military trying to freeze dry red blood cells so they can load like Tralos into red blood cells. Then you could have powdered blood done stable at room temp forever. But, all right, I want to talk a little bit about what we're doing today in this journey that has got to here before I run out of time. And I say feel free to interrupt as we go. But I'm now focused mostly on longevity and with a new kind of tool. And I mention all that stuff in the back because it's woven itself into what we're doing today. So I've developed kind of a professional obsession with nucleic acid delivery, and then still really pursuing this idea of inflammation in life as a tool to mitigate disease and age. And so here for Ocean, our primary lab is down in San Diego. We're actually in the same space as a company called Entos, which developed the delivery technology we're using. And I'll tell you a bit about that. And so I say I'm fluent in English and gibberish. Feel free to push me in one direction or the other if you want more technical details or less. I can go back and forth. But the general idea here is that age is just the master corollary for every malady you can think of. I'm sure you've probably seen a million versions of this graph, but the results are always the same. Like the older you get, the more screwed you are. And I mean, far worse odds of having cancer is like a clean living 70-year-old than you do a chain smoking 20-year-old. And it's not to say your ill-spent youth won't catch you later. It's just that that age itself is such a dominant factor that it drives the others. And so we wanted to go after the things that really drove aging, so we could build therapies that would address a lot of things. And as a company, though, we're forced to kind of toe an interesting line in that we still need to make things that like pharma would buy and the FDA would approve. But FDA doesn't think aging is a disease. And for most of history, aging therapies have been viewed as like ground zero for snake oil. And so it's this challenge to find what things can we like actually target that will have a meaningful impact on this in the short term and allow us to build things that would have a much more profound impact over the long term. And it's back on the same thesis that you're never going to be able to take enough pills to fix aging. So we need to get into the code itself. And when we put this company together, we didn't just look at what is a treatment we could make today or what's a therapy, but more of where's the field going to go in the next 20 or 30 years? Because we were looking at some of these things. I'm going to talk a bit about like building muscle, killing fat, our senolytic stuff. Those things were things that we could do relatively quickly, but others like the science wasn't really there yet and still isn't. But our bet was that as the field progressed, they would still need a way to deliver this code. And so we wanted to build an empire, much more of a big pharma for longevity than like just a single asset we would flip. And so we started out looking at these kind of common problems of like muscle accumulation of unwanted fat and senescent cells. And I'm going to talk a little bit about what we're doing with those, but I'll go through the platform very briefly and kind of contrast it against what we've done in the past. And so with both Immusoft and Sigma, they had to have cells out of your body to modify them for practical purposes. The in vivo electroporator would work in a person, but you can only get like a small amount of tissue. You couldn't like just, do it to your whole body. And so for longevity though, we wanted something that could go after basically every cell in the body. And there's technologies that have become fairly dominant to do this, viral vectors and non-viral vectors like LNPs. And they both have like a bunch of problems. I mean, on the virus side, they're obviously very good at delivering nucleic acids, but your body knows what viruses are and doesn't like them. It's very easy to get immunity to them, and they tend to have limited payload capacities, at least for the ones most commonly used. And the LNPs, which everyone knows now from COVID vaccines primarily, but they are very relatively cheap and easy to make, but they can't deliver DNA for practical purposes, and they become wildly toxic when they accumulate. And so we wanted to find the best of both worlds to this. So like the cost and structure of a lipid particle with some of the entry characteristics of a virus. And so we have this thing called a proteolipid vehicle, or a PLV, and it basically will fuse with the membrane of any cell it comes in contact with. So notably, it doesn't have to be endocytosed. It doesn't need to be swallowed up by the cell. And that's like a subtle difference that matters a lot. And it's what allows us to get away with like very high doses of things and not triggering this like really powerful innate immune response. And it was made possible by something I could nerd out about for a lot, but I'll spare you right now, at least until Q&A. And this little fusogenic peptide. And so this was first discovered from a fusogenic reptilian orthoreovirus, a virus that infects the stomachs of alligators. And it's really interesting for a couple of reasons. One is it's the only non-enveloped virus known to science that has a fusion protein. And it's about 100 times smaller than the next fusion protein that's capable of fusing membranes. And so it's, from a like evolutionary point of view, wildly different than anything else that has ever been seen. And early on, scientists got this idea of, put this on a lipid particle. That's got to help it get in, and it does. And what's amazing is it doesn't need to be swallowed up anymore. It can just fuse with the membranes. It'll mix the lipid of the cell with the lipid of the particle and deliver it. And this allows you to administer doses that are crazy high without toxicity, and do things that you could never dream of with an LNP. Where like if you did this with a normal LNP, the liver would have 90% of it at least, and basically there'd be nothing else. And when we started this, so my goal was to hit every cell in the body, have a flat biodistribution curve and no tropism, which is a kind of an absurd ask if you're familiar with that space. But here, unless the liver is down here, like below the heart and the bone marrow, it's no longer a primary uptake tissue. And that's because this particle is not made of cholesterol ionizable lipids. You can tailor its distribution really radically. And I don't have slides for that in my main presentation, but I could show them to you later if you want. We can skew this to go after pretty much any tissue we want. But the idea behind it, though, and why we were doing that is, I think, best illustrated here. So in this case, we have a mouse that got an IV injection of the PLV carrying DNA that makes firefly protein, luciferase, just a reporter. And it's under a promoter that's on everywhere. So the whole animal basically lights up. And if you chop it up and look at the tissues, they're all lit up also. But this one on the right's the more interesting one. So it got the exact same dose, the same route of administration. So IV injection, still the same amount of DNA. But now that firefly protein is being controlled by an engineered promoter, a logic gate of sorts that was designed with DNA, and a tumor is implanted inside this red dashed line. And so now it's being told to only turn on in the tumor. And you can see that the expression is totally restricted to that. And all these tissues got the same amount of DNA, but it's not turning on in there. And I think this is a really important distinction, that like every nucleated cell in your body has the same genome, but you're kind of running different apps in a muscle cell than you are in a neuron or a skin cell. And so this control logic of life, which I think is the most fascinating part of all, is the way we're targeting this now. So we're able to like say, I want to pick off one bad cell in like an ocean of good cells and not have collateral damage. And the only way to do that is to not use a drug at all. It's a little computer program written in DNA. All right, so I've already taken longer than I thought. So I'll go quickly through what we're doing, and then we'll switch over to normal conversational mode. So the first one is building muscle. The reason we're doing that is because physical strength is everything as you age. I mean, not just resilience against disease and injury, but metabolic health, cognitive function, everything's tied to it. And the genetic control systems for muscle building are actually pretty well understood. That monstrous cow there is not photoshopped, and it was first characterized in the 1800s. And so there's the pathways, follistatin, myostatin, activin, that are regulating the growth of muscle fibers has been studied for a long time. And if you, in fact, overexpress the growth factor, follistatin, you get an animal that's 4 times the natural size. So it'd be twice the size of this cow if you could do it in a cow, which would be unpleasant for its mother, I suspect. But the pharma's been trying to make drugs for this forever, and for decades, literally. And we think we can use this technology to succeed where the others have failed in the past. And so this is an example of what we're doing with our treatment. And we gave these mice a single administration of this, and in 6 months, they're 50% larger and twice as strong. So you're not making like the super roided out creature anymore, but and I think the most anyone's done in a primate with a tool like this is about 30%. But I don't know, I weigh close to 200 pounds, so 30% increase would be really substantial. But so in really old animals, when we've done this, they don't get actually super big, but they do get statistically stronger. And so I think this could be a treatment that we could make affordably enough that the payers would be basically pushing the patients to do it, because it'd be cheaper for you to be strong than it would be for you to break your hip for them. And you can also inject it in a single local administration and use it for cosmetic purposes, like build bigger chest or butt or something like that, which I think there's interest from in the pharma world. On the fat side, we were using that same targeting method I showed you earlier with the reporter to kill a fat cell selectively. And we're taking it for lipedema, so another rare disease that's characterized by abnormal fat deposition. And the only real treatment for it is repeat liposuction and lifelong ketosis. And so I think you could basically do body sculpting with a hypodermic needle, but now or you wouldn't have the trauma or the bleeding. You could just make the cells die, and it would be accepted well in hopefully a lucrative treatment that would allow us to move into things like trying to kill visceral fat, which is where I'd like to go. But that's all on that one. I'll leave it on this. The original proof of concept from the transgenic fat killing in animals is actually predated some of the senolytic studies done by the Buck and the Mayo Clinic. You're familiar with those. The Ink attack model used to be a fat attack model. And they could wipe out every living fat cell in the animal in 3 days, take this huge fat mouse into a skinny mouse. And I think incidentally it's a terrible idea for doing this to a human. I don't know if they'd survive it, and it would certainly be bad cosmetically. You'd have like a skin suit, basically, wings. But I have it in here because it shows they still weren't hitting any of the wrong tissue. That logic gate was still sound enough to only kill the fat cells. And so even though you wouldn't do it on a broad sense, you could do it in a local. And actually, I'll show one piece of eye candy before I stop talking for now. So this is done as a earlier kind of in vitro proof of concept, and we're trying to kill senescent cells. So we want to kill P16 positive cells, and we made this vector that would make the cells express GFP, so we could see them, and then we could kill them with a suicide gene. And you could activate that suicide gene and watch the cells basically die before your very eyes. So it's a time-lapse microscopy over a few hours, and the cells are going into apoptosis. But what's really amazing about it, more valuable really, is that if you have a mixed population of cells where only 5% of your cells are the target, you can do the same thing and not hurt the bad ones. And so this is what allows a level of precision in targeting that you really couldn't do any other way. And so that's kind of the tool chest we're playing with right now, and building all sorts of fun stuff. And I'll stop for now.